Hey everybody, Mr. Murphy here. I'm going to go through chapter 9.2, which is all about DNA replication. I'm going to cover this kind of quickly, and you can go to the other video I have for some of the details about leading and lagging strands and all the different proteins involved. But for this, I'll keep it kind of big level. So first off, DNA replicates semi-conservatively, and what that means is that the original two sides of the DNA molecule split, and then we match those up with their complementary bases. And so each of the two new cells that are being produced are going to have one side of their DNA that came from the parent cell. And so that came through experiments that we've talked about in class, and you can look into those. They're very interesting. So the two big steps here is that the DNA, the double helix needs to be unwound, and it basically needs to be unzipped. And then the new nucleotides need to be matched up to the two sides that we've separated there. So you could think of this as you have one set of DNA, you unwind it and separate it, and now you have these two templates. And they're complementary to each other because we just peeled them apart. But what that means is that each of them is going to code for or inform what the matching thing that needs to be added on. And by the time you're done replicating the entire length of these two separated strands, you will have two identical copies of that DNA. So the template DNA is read in a 3 to 5 direction. We talk about that with DNA polymerase, which is the star of the show. That's the main enzyme that's responsible for following along the single-stranded, separated strand of DNA, and it follows it in a 3 to 5 direction, creating the complementary base pairs. There's a good bit of additional information here about other components that are specifically related to this. Just know that the DNA replication, the other video that I have and what we've talked about in class, is what we want to primarily focus on. So while there's a good bit of some extra information and structures like mentioned here, these are not relevant to what we specifically need to cover. So if we look at this, we have each of the template strands, or this template strand here, that is having this growing strand matched onto it. So the template strand is being read in the 3 to 5 direction, and what's happening is we're matching these up one at a time. And so here you can see a cytosine being brought in to be matched up with this guanine, and that happens here. Excuse me. And when that happens, these free nucleotides that are available in the environment of the nucleus have these phosphate groups that are present, and those will get broken off. Two of those will get broken off, and so that phosphorylation is involving ATP. So somewhere else separate from this, we have a process that's generating the available nucleotides in some energy requiring fashion. So these are basically prepared, and the nucleus is chock full of these when the S phase of interphase occurs. And so just to understand that there's a chemical process to basically grabbing the available thing, breaking off this piece, and attaching it in. And you can see how that one phosphate group becomes the part of the phosphate sugar chain. So DNA polymerase, I mentioned, is the whole the thing that's involved here, and we have the point at which this begins on the DNA strand is called the origin of replication. And what happens is, if you imagine a DNA strand here straight across, this origin of replication is where we begin that replication process, and it sort of makes a bubble in the DNA strand, and then we have two replication forks that extend in either direction, and that's basically like the zippers pulling apart in either direction. So you'd have this happening in two directions, and you can have multiple origins of replication. And so you could have this happening, like here's a plasmid from a prokaryotic organism, but you see this is where we pop open the middle, and then it begins to unzip to the left and the right. But you could actually have multiple of these ories extending to the left and right in a single strand, and they'll continue until they meet here in the middle and they separate fully into two now replicated strands. It begins with a primer needing to be placed by an enzyme called primase, which puts down a little RNA primer on the single-stranded DNA, which notes a place where DNA polymerase can then grab on and then begin its process of adding the nucleotides. And so here we have a separated strand. So this is like DNA strand that's been separated out. A little primer of RNA gets placed. Polymerase grabs on there and then begins making the new complementary strand. There are other components then that step in and basically need to address various concerns, including the removal of that primer. This talks about how we can think of the DNA polymerase as being like a hand, and like a hand, it has a very specific shape that it can grab onto the DNA with, and that's why it has to follow the DNA 3 to 5. It can't grab onto it and go the other direction because it just doesn't fit into the shape of the DNA polymerase. Which, again, a concept we've talked about a lot. Proteins have such specificity to their shape that it might seem real easy that you could just have it go in both directions, but it just doesn't have the right shape to fit that. And for that reason, we need to think about the fact that DNA is anti-parallel, and so that means that we're going to have kind of two different things happening on either side of the DNA molecule. Don't get confused by statements like this. DNA replicates in a 5 to 3 direction. The DNA polymerase follows 3 to 5, but that means it's creating 5 to 3, and that can get really confusing. So I would really just commit the one statement of DNA polymerase is following the template from the 3 direction to the 5 direction, and everything else you can just figure that out as needed. So here you can see the primer's put down, DNA polymerase is going, but this creates a problem because we can't on this side do the same thing. We can't put the primer down and have DNA polymerase go to the right because that's the 5 to 3 direction. That's the wrong direction for it to be following. And so this creates the need for the lagging strand, and these discontinuous little chunks of the lagging strand are called Okazaki fragments, named for the individual responsible for sort of figuring this out and piecing this together. And so here we have the leading strand up here, the lagging strand on the bottom, and the lagging strand is made up of several little segments because as it unwinds more, a new primer gets put down, a new polymerase comes and begins replicating to the left here, and that creates then this next little segment, and then more gets unzipped and another primer gets put down and another polymerase comes and begins writing it. And so that's necessary in order to create that other side of the DNA that needs basically more material to be unzipped and revealed in order for the polymerase to grab on and to be able to do its job. So this creates one issue, though, which is that these little segments don't get attached together. Polymerase is so specific in its job that when it runs out of single nucleotides to be matched up to, it just stops. It doesn't have a way to like attach to the very last thing. Not to mention that this little green piece here still is an RNA primer potentially. And so DNA polymerase 1 comes in, a different DNA polymerase comes in and replaces that primer. And then we have a last enzyme called ligase that comes through and basically just stitches up those little segments and makes this one continuous piece. And so that's important for the final sort of fixing this gap, and we have one now nice piece of DNA. Okay, so here we see this is DNA polymerase has reached the primer, and then that goes away. And actually, this is not ligase. This is the DNA polymerase 1 coming in and replacing the primer, and then we have ligase comes in and fixes the little snipping here that's left behind. Okay, so it's a process of primase marks it for the DNA polymerase that comes in and makes it, and then DNA polymerase is ends its job in a different polymerase called polymerase 1, comes in and replaces the primer, and then the DNA polymerase 1 is done, and then ligase comes in and actually stitches it. And that is that's the process. So this is very fast. It's like 2,000 to 4,000 nucleotides per minute is how fast DNA polymerase can operate. And because it is happening in multiple places along the length of each of your different chromosomes, it is happening very, very quickly. Okay, so this last little section here gets into the fact that because the last primer gets removed, we lose a little bit of our DNA each time this process happens, and so these cut little segments can become problematic over a long enough period of time, because while our DNA has a bunch of sort of extra filler pieces called telomeres at the end that are okay to lose, as you lose more and more and more, you might begin to lose relevant coding pieces, and so that becomes an issue. But because of the actual fitting of the enzymes onto the end of the DNA, it's not possible for that last piece to sort of get properly replaced. And so that's why this is something that occurs. So telomeres help to prevent that by just being extra junk DNA at the end, these repetitive segments that we can stand to lose. And then there's an enzyme called telomerase that helps to sort of restore those telomeres, but it is not a perfect process, and so there is always a loss of that DNA over time. And then let's see. So we'll get into this a little bit more with the mutation section, but there is a process of proofreading going on. There's a process of, whoops, this A shouldn't be here, and it's creating a problem because it's not properly hydrogen bonding with this G that's across from it, and flipping out the one nucleotide for a new nucleotide, and stuff like that. So that process is also happening, sort of swarming around in the background of this whole thing, making sure that the overall process is correct. Here you can see a mismatch repair, and some protein would basically remove that, and then polymerase would come through and match it up again. So while we like to think of this as this very ordered process, it really is lots of things doing their job, sometimes multiple times. If pieces get removed, then polymerase has to come back in, and then ligase has to come back in, and so it can be this whole repetitive process. But in the end, because it's happening on the chemical level very, very quickly and in many, many places all at once, it's relatively very fast and relatively very efficient in the end to produce 2 new copies of DNA. PCR was one of the technologies that we talked about, which is a way in which we can basically go through this exact process, but in a controlled fashion. And so what you need is a double-stranded DNA sample, and then you need some primers and some other molecules that will help to basically identify, tag, and cut the DNA segments into what it is that you actually want. But from there, basically what's happening is by raising and lowering the temperature, raising the temperature to separate these 2 strands, lower it, add in a primer, add in some polymerase, and then we get 2 replicated copies, or we get a replicated copy, 2 copies of the DNA. And then you heat it up again to separate this, and then you cool it down, and you do polymerase, and you can just make tons of copies of a sample of DNA. And so this is a process we use today for a whole bunch of reasons, including COVID testing and forensics and all sorts of good stuff. That's the end of 9.2. So the next one is all about mutations.